السلام عليكم ورحمة الله وبركاته. أعزائي طلاب الثانوية العامة شعبة علم علوم، أهلا بكم في حلقة المراجعة النهائية. This episode very important for you. Why? Because it focus a main point that help you to solve MCQ multi-choice questions. I tell you instructions about the final exam. إن شاء الله. إن شاء الله توزع عليك البابل شيت، تمام؟ اللي أنت بتظلل فيه بإذن الله، ومعاك المودلز سواء كان A or B or C. تمام. أنت بتعمل فوكس جامد جدا على المودلز اللي قدامك بحيث إن أنت تقدر تفهم السؤال جيدا. Please focus اللي understood the questions please. Why? Because it's very important. That's help you to solve to choose A or B or C. اوكي. بعد كده ان شاء الله باذن الله تعالى تختار اجابتك موجوده في المودلز اللي قدامك. فاينلي خالص المرحله النهائيه باذن الله تعالى حضرتك بتمسك البابل شيت وتظلل. ليه؟ لان انا اهم حاجه عند التصدير يكون مطموس جيدا. ليه؟ لان في برنامج اسمه ريمارك، البرنامج ده احنا بنعمل عليه فوكس قوي، ده اللي بنصحح منه. طب لو حصل مثلا سي مثلا ان واحد ظلل مثلا وجي مسح بعد كده، ممكن الاجابتين الاثنين يكونوا مطموسين، مش هيقراها ريمارك، في الحاله دي بتبقى فيه مشكله وحاجه فيها ديستربانسز، وده عليك خطا كبير جدا. فلازم نقرا المودلز كويس جدا، نختار اجابتنا من المودلز، وبعد كده في الاخر خالص نظلل اللي هو الفاينل اللي هو الايه؟ فاينل انسر بتاعتي. اوكي. Now we are going to the first part, movement and support through the first questions. Follow me. Okay. Number 1. Which of the following cell acquired a physiological support on watering the plant? Number A, sieve tube. B, xylem vessels. C, stone cells. parenchyma cells. احنا قلنا قبل كده الفسيولوجيكال support depending only in parenchyma cells and collenchyma cells. Parenchyma cells is contain the living cells, so it's play an important role in the physiological support. But stone cells, it's not considered from physiological support because non-living cells. It's called sclerenchyma. It contains both of cellulose and lignin. So the best answer, it's called parenchyma cells. Why? Because the parenchyma play an important role in a physiological support. That's right. Second questions. Treasure pressures arise in the plant cells under the main effect of A, cell wall. B, plasma membrane. C, cytoplasm. D, cell vacuoles. Please pay attention about the treasure pressures. There is a inverse proportions between the treasure pressure and osmotic pressures. Treasure pressure is arising when the self vacuoles contain a large amount of water. So the concentration of water is increased and the concentration of the soil is decreased. So the amount of water is pressured into the plasma membrane, so it's called treasure pressures. This is inverse proportions between the osmotic pressures. Then the osmosis pressures is contain a large amount of solute, decreasing the amount of the waters. Okay, so it's called inverse proportions between the treasure pressures and osmotic pressures. So the best answer, it's called self vacuoles. Why? Because the self vacuoles, it contains the solute and solvent. Solvent, water, and solute, اللي هو الايه؟ السولت او الايه؟ السugars. That's right. Other questions. The opposite graph illustrating the changing in the size of self vacuoles in the epidermis cells of a flowering plant leaves that was put in a solution with a different concentration through a certain time period. Number 1, what is the least concentrated solutions? A, X, B, Y, C, Z, D, L. The best answer L. Why? Because the size of self vacuoles is high. Okay, why? The size of self vacuoles is bigger. Why? Because the self vacuole it's absorb waters. The waters it transferred from low concentrations of water, low concentration of solute to low concentration of solutions to high concentration of solutions, or passing the waters from high concentration of waters to low concentration of waters. So the best answer it's called L. Number 2, what is the highest concentrated solutions? Highest concentration of water at X, Y, Z, or L. The best answer, why? Because the size of the vacuole decrease. That means that the waters passing from exostasis, exostasis. تمام؟ الwater is passing from inside to outside, so it's called losing amount of water due to the solute inside the cell vacuole. It is the low concentrations and the water passing from inside to outside, so it decreases. That's right. Other questions. Which of the following is correct about the last two groups of the vertebral column vertebra? Vertebral column, it consists of 33 vertebrae. Beginning with the first group, it's called cervicals. It consists of 7 vertebrae. Second group, it is called thoracic. Okay? It contains 12 vertebrae. Third group, it's called lumbars. It contains 5 vertebrae. First group, second group, and third group, it's called articulating. That means that articulating يعني متوصلين بيتحركوا. The last two groups, sacral and coccygeals, it fused, so it's considered 2 bones. Also, the sacral, it contains the 5 vertebrae, but it fused. It's considered 1 bone. And the coccygeal, it contains 4 bones, but it's considered 1 bones. Why? Because it's fused. Okay? So the similars, they are similar in the number of bones. Okay? The last two groups of vertebral columns, mean sacral and coccygeals, it's contained as a similar number of bones. Sacrals, 5 vertebrae, but fused. Coccygeals, 4 vertebrae, but it fused. So it is similar in number of bones. It considers number of bones, and another coccygeal, it contains number of bones, so it's similar to the number of bones. This is a piece right. Another questions. Okay. What is the number of the depressions in the appendicular skeletons? A, 2, B, 4, C, 6, D, 8. Okay. Pay attention, please, in this point. There is an appendicular skeleton. It's contain upper limb and lower limb. Upper limb, it's contain upper limb and the pectoral girdles. Lower limb, it contain the lower limb and the pelvic girdle. In upper limb, in the outer board, in outer end of the scapula, it is called the glenoid cavity. 1 cavity and another cavity. Okay, 2 cavity. In radius and ulna, attaching the humerus with radius and ulna, it's called in cavity, 2 cavity. 4 cavity. Acetiblium, it's found in the pelvic bone, it's 2 cavity. The total number is 6 cavity. Okay? 4 in upper limb and the pectoral girdle, 2 in lower limb, that's found in the pelvic girdle. So the total number is 6. This is a piece to answer A, C. It's called 6 cavity. That's right. The connection of the 3 lumbars vertebra with each other required the articulations of space process. 4, 8, 12, or 16. Pay attention, please. There is the 3 vertebra. For example, vertebra number 19, 20. Okay? And the lumbars beginning with 20, 21, 23, 24, 25. Okay? Vertebra number 20, for example, 21, 22, 23. Okay? 21, 22, 23. 23 ascendingly is connecting with the 22 anterior articulating process. 22 connecting with the 23 posterior articulating process. يبقى أنا هنا عندي 4. Okay? 23 is connecting 21 anterior articulating process. يبقى 6. 21 connecting with the 22 with the posterior articulating process. The total number, it is 8 arterial articulating process. The best answer, it's called 8. Why? Because it is the anterior articulating process with the posterior articulating process alternating with each others when the connecting 2 bony vertebra with each others. That's right. Another questions. That's right. Myofibrils consists of 5 simulated band H, 6 darked lines Z, and 5 darked line band A. So how many sarcomeres are present in the myofibrils? Pay attention, please. The muscles fibers, it is a structures unit in the skeletal muscles. Okay? Skeletal muscles is surrounded by a group of bundle muscles, surrounded by bundle membranes. It contains multinucleated. Why it contains multinucleated? Because the nucleus, it contains the nucleus. That's a ribosome, it's made inside it. So the ribosome play an important role in the formation of the protein, especially for myosin and ductin, the main component of the myofibrils in the structure unit of the muscle fibers. Okay? Muscle fibers, it contains myofibrils, 1,000, 2,000 myofibrils. It ranging between 1,000, 2,000 of myofibrils. It divided in two. Light band I, it's bisected by darked line, it's called Z. Okay? Darked band, it's called A. It's bisected from inside with the semi-lighted band H. During the contraction and relaxations, the light band A, it is decreased, and Z approach to each other. The sarcomere is decreased. During contractions, the light band A, it's stable, and H may be disappear, may be decreased. Okay. This is my fiber consists of semi-lighted band H, okay, 6 darked line Z, and 5 darked band A. So how many sarcomeres are present in this myofibers? The number of Z lines equals number of sarcomere plus 1. Light band I equals sarcomere plus 1. Darked band A equals the number of sarcomeres, and equals the H band, the semi-lighted band H. So the number of sarcomere is called 5 because it's 6 darked line Z. Minus 1, it's called 5. So the number of sarcomere in this point is 5. That's right. From the concentration of the skeletal muscles, the presence of the necessary lactic acid and choline, ATP and calcium ions, ATP and choline, acetic acid and sodium ions. Okay, please pay attention to me. During the contractions of the muscles, okay, during contraction, you need the calcium ions. Why? As soon as the nerve impulses, transmissions from the motor nerve cells into myofibrils that's found in the muscles fibers, there is a synapse that contains neurochemical transmitters. As soon as the nerve impulse is passing through it, the calcium ion enter to the synapse, ruptures the synaptic vesicles that contains chemical transmitters. It's called acetylcholine. As soon as the acetylcholine is ruptures, the muscle fibers charge is changing from polarization in depolarizations, helping to the muscle contractions. So we needing the muscles contractions, ATP and the calcium ions. As soon as the muscles relaxations, need ATP and the cholinesterase enzyme. Why cholinesterase enzymes? Because the cholinesterase enzyme play an important role in the compositions of the acetylcholines and acetic acid and the cholines. So the muscle returning to the normal positions is called relaxation. Pay attention, be in this point. As soon as the cholinesterase enzyme decomposed the acetylcholines and acetic acid and the cholines, the channel of the sodium is closed and channel of the potassium it's opens. The amount of potassium outside the cells, it's more than inside the cells. After a few seconds, the channel of the sodium it's open, and sodium outflow outside. The outer surface membrane is called positive and the inner surface is negative. So the muscle retaining to the normal positions at rest. So the muscle it relaxations, and the amount of potassium ions inside the cells is more than outside the cells. That's right. Another questions. Which of the following statement is not correct about the hormones? The concentration of the hormones could be regulating by another hormones. That's right. Hormone it concentrations regulating by another hormones. For example, thyroxine hormones, it's stimulation by effect of the TSH, thyroid stimulating hormones. Okay, that's right. That's right. B, the concentration of the hormones may be have stimulating or an inhibitors. That's right. Okay. TSH hormones may be stimulation the thyroxine hormones to helping to the secretions to make a play important role in the rate of the metabolic malleability of the skin and hairs. If the TSH hormone it decreases, it inhibits the actions of the thyroxine hormones. So when TSH hormones it's high and thyroxine is high, the disturbances it's occur inside the thyroid gland. That is when the TSH hormones is secretions is hypersecretions and the thyroxine hormone hypersecretions, the disturbances it's occur inside the anterior lobe of the pituitary gland. It's called adenohypophysis. When the TSH hormone hyposecretion and thyroxine hyposecretions, the disturbances it's occur in adenohypophysis in the pituitary gland. When the TSH hormones, it is the hypersecretions, and thyroxine is lower secretions, the disturbances inside the thyroid gland. That's right. Another questions. Okay? 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 That's right. Another questions. Okay? When drinking a large amount of waters, this lead to the drink a large amount of waters. That means that the osmolarity of the blood it's decreased. So the ADH hormones that's secreting, not, please pay attention, not secreted, but it releasing and the storage inside the posterior lobe of the pituitary gland. It's called neurohypophysis, but it's secreting from the neurosecretory cells that found in the area. It's called hypothalamus that found in the brains. When drinking a large amount of water, this leads to the increasing in TSH hormone secretion. It's wrong. B, increasing in ADH hormone secretion. No, because the osmolarity of the blood it decrease and the osmolarity of the urine it's increase. C, decreasing in TSH hormone not related to this point, but the best answer is D. Why? Because decreasing is the VH at the same meaning of the ADH hormones. It's called antidiuretic hormones. It's secretion. That's right. Another questions. Which of the following process is stimulated by the hypersecretion of thyroxine hormones? Increasing the production of ATP molecules. Why? Because the thyroxine play an important role in the metabolic. Metabolic is divided into catabolic, oxidation of the glucose, producing a large amount of ATP. ATP, it's considered storage on energy. So it's called a standard universal currency of energy inside the cells. When it released, it transfer into ATP, releasing the amount of energy from 712 kilocalories. That's right. Another answers. Decreasing the speed of the nerve impulses transmission is not related to. Increasing the glycogen amount that is stored in the livers, not related to. D, decreasing the sodium level in the blood, not right. But is the best answer A, increasing the production of ATP molecules. Why? Because the thyroxine play an important role in the regulation of the carbohydrate in the blood and rate of the metabolic. It's producing a large amount of ATP. That's right. Okay? The hormones that affect the metabolism of the carbohydrate are secreting by the effect of space hormones. That's right. ADH, not related to that, but the ADH is related to the osmolarity of the blood and the osmolarity of the urine. FSH, it's affecting the ovary and affect the testis. In ovary, producing the follicular graphians that's secreting estrogen hormones. Testis, seminiferous tubules that play an important role in the spermatogenesis. LH, luteinizing hormones forming the corpus luteum in ovary and forming the interstitial cells that secreting androgen, testosterone, and androsterone, not related to. But ACTH, adrenal-corticotropic hormone, it related to the hormone that affecting the metabolism of carbohydrate. So the cortisone, it's called glucocorticoid, that's secreting from adrenal gland cortex. It's stimulated by the anterior lobe of the pituitary gland. It's called adrenal-corticotropic hormone. Okay, that's right. إن شاء الله نخرج فاصل الآن ونعود الاستكمال الحلقة النهائية للمراجعة النهائية لمادة الأحياء باللغة الإنجليزية. السلام عليكم ورحمة الله وبركاته ونعود بعد هذا الفاصل. السلام عليكم ورحمة الله وبركاته. أعزائي طلاب الثانوية العامة شعبة علم علوم، رجعنا لكم مرة تانية لاستكمال حلقة المراجعة النهائية لمادة الأحياء باللغة الإنجليزية، وإن شاء الله أكون ضيف خفيف عليكم، وإن شاء الله أقدر أفيدكم بإذن الله تعالى، موفقين بإذن الله تعالى. نكمل مع بعض. Insulin working on the transferring the glucose from blood, from cells to blood to urines. Insulins transfer the glucose from blood into cells to oxidize to make the catabolism to producing ATP. That's right. The best answer is from blood, not to blood. That's right. Another questions about the hormones. Which of the following hormones affect the digestion process of a piece of bread? Gastrin, it related to gastric juice that's secreting from the stomach, related to proteins. Thyroxines, carbohydrates, and play an important role regulations the carbohydrate in the blood. Insulin transfers the glucose into the blood. Cholecystokinins, that's a bestrite. Why? As soon as the food reaching to the duodenums, the mucous gland lining the duodenums make secretions, cholecystokinin and secretin, stimulating the pancreas, secreting the pancreatic juice to digestion the bread or starch into a amylase. Okay, that's right. Into maltose. That's right. Okay, another questions. Which of the following is used in a tissue cultures technique? This is be attention please about the reproduction in living organism. Tissue cultures. When you make a tissue culture, this method it's called artificials, not naturals, producing an identical living organism from the parent cells. Carrots take the tissue and putting in semi-nutritive medium, it's called coconut milk. Why? Because the coconut milk, it's contain auxins, play an important role in the stimulation to growth. So it grows in the seedling and putting in the land to reaching the new individuals in the fast way, so it's solving the food problem. The best answer is parenchyma because the parenchyma is living cells. Sclerenchyma, non-living cells. Sieve tubes of phloem, non-living cells. Xylem vessels, non-living cells. So the best answer is parenchyma because it's living cells. That's right. Another questions, according to the reproduction of living organism, which of the following its presence is necessary for completing the asexual reproduction in Hydra? Interstitial cells is the best answer. Why? Because the interstitial cells play an important role forming the bud. The bud it grow forming a new individual that contain the same genetic materials that found in the parent cells. So the best answer is interstitial cells. Another question. What is the number of the complete meiotic divisions that occur on the germination of zygospore in spirogyra algae? The number of meiotic divisions when the conjugations it's occur between two filaments adjacent to each other. That's right. The protrusions it's appear and the conjugation tube it's connections between two filaments. One filament protoplasm transfer to another filament forming the zygote. It contains 2N. The zygote surround itself by the cutaneous membrane or thick membranes. It's called zygospores. Zygospore it's dominate. Okay, the number of chromosomes that found zygospore is 2N. Dominate under the suitable conditions, it's improved. That means that this division is conditional. What the meaning of conditional? Mashrut. When the condition it's improvement, the divisions it's occurs. So the best answers 1 divisions it's okay to producing a new 4 cells, 3 degenerate and 1 is disappears and complete 3 degenerate and 1 it's appear. 3 degenerate, 1 appears, 1 is complete mitotic division forming a new filament and from this mitotic divisions in the scleroformation conjugations or lateral conjugation in the spirogyra. It's specific type of sexual reproduction. It's occur in unsuitable conditions. Another questions. Meiosis, space, the conjugation, and the space is the fertilizations. Meiosis precedes the conjugations or follows the conjugations? I think it's follow. Why? Conjugation it's occur at the first and meiosis it's occur. يبقى first space it's precede يعني يسبق and fertilizations that mean that اللي هي ايه بناء على اسمها مين اللي هي ايه precede. يبقى number 1 meiosis follow the conjugations. Number 1 meiosis follows the conjugation. After the conjugations it occurs the meiosis it's occur. So it's called follow. And the space that found in the fertilizations, before the fertilizations, the gametes is formed by meiosis. So it's precede. It was the best answer, follow and precede. Follow conjugations and precede the fertilizations. That's right. What is the infected phase for the female Anopheles mosquitoes in the life cycle of malaria plasmodium? The infected phase, okay, focus again in these questions, please. What is the infected phase for the female Anopheles mosquitoes in the life cycles of malaria plasmodium? Sporozoites, this is infected phase for the humans. Gametocytes, that's right. When the sporozoid is transferred into the blood, reaching to the lymphatic cells, it divided mitotically by schizogeny, producing a large number of the merozoites. It's ruptured from the liver cells, attack some type of red blood cells. When attacks some red blood cells inside some type of red blood cells, okay, it divisions it's okay, producing a large amount of phenyzoids every 2 days and releasing by the toxic substance. This phenyzoids is transferred into immature gametocytes. When the healthy female Anopheles mosquito petting the infected human with the malaria, the immature gametocytes, it differentiations and maturations inside the stomach of the female mosquitoes as transferred into male gametes and female gametes to complete sexual reproduction and life cycle of the plasmidium. So the best answer is called gametocytes. That's right. Another question. Which of the following structures in the life cycle of the plumediums formed by meiosis? Spores, that's best right. Why? Because the spore is formed by meiosis. Oviums is formed by mitosis. Seriated sperm, as the same, by mitosis. Sporophytes, by mitosis. Through the zygotes, it's okay, the fusion between male gametes and female gametes. In the zygotes, it's followed by the mitosis to complete forming the new individuals. That's right. Another questions. According to the reproduction of living organism, pay attention, please. What is the maximum number of tube nuclei in the pollen grain that are result after the germination of flowered anthers, where each sac of it contains 120 spore mother cells? A, 48. Pay attention, please. Each answer it contains 4 chambers, 4 غرف. Okay? Spore mother cells is divided mitotically, forming the pollen grain. Each spore mother cells producing 4 pollen grain and producing 8 main nuclei. But that means that each spore mother cells producing 4 pollen grain and 8 main nuclei. 1 main nuclei, 4 main nuclei. In each 4 sac, 4 by 4. Okay? The number 120 by 4 equals 48. 48 by 4, the total number 1920. So the best answer is C. It's what? 1920. Okay. Other questions about the reproduction of living organism. In which is the nucleus similar to the endosperm? Nucleus lining is the embryonic sac from inside. Nutritive mediums. Endosperm, nutritive medium, especially for the exoendosperm. For example, dicotyledons, using the spore to feed on oat. For example, beans, pea, lupins, apples. Okay, so the best answer is the function. Both of them play an important role in nutritive medium. That's right. Another questions about the reproduction of the living organism. In which of the following plants does the embryo feeding on the endosperm during the development? Okay, onion, monocotyledons. Maize, monocotyledons. Apple, dicotyledons. The dicotyledons, it's feed on the endosperm. So the best answer is apples, not palm. Palm, it's considered monocotyledon. Another questions. Reproduction of living organism. In which of the following stage the chromosomal content of the cells doesn't change during spermatogenesis? Multiplications. Multiplications, that means that the primary germ cells are divided mitotically. Mitotically producing a large number of the spermatognia. The spermatognia, it producing oogenia. Okay, sorry, the spermatognia, large in size, it's transferred into primary spermatocytes. This is no change, okay? And the growth stage, best answer, multiplication and the growth. No changing in the number of chromosomes. So in which of the following stage the chromosomal content of the cells doesn't change in multiplications and the growth stage? That's right. B, growth and the mutation, they change. Multiplication and the mutations, it change. Mutations and the metamorphosis stage, it is changing. So the best answer, multiplication and the growth stage. Okay? What is the number of female gametes that are resulting from 5 primary oocytes? Each primary oocyte producing 1 secondary oocyte and 1 polar body. When the females, it is not married. After the female had married, producing the 3 polar body and the mature ova. Due to the filtration, it's okay in anterior first third part in fallopian tube. So the best answer is 5. Why? Because each primary oocyte producing 1 secondary oocytes that releasing in 14 days from the beginning of the menstruation. It had ready to fertilizations when meeting the sperm by secretions, the hydrodase enzymes that secreting from a chromosome that found in the head of the sperm. That's right. Okay. What does graphial follicles contains on the fifth day from the beginning of the menstruations? Already primary oocyte. After that, it contains secondary oocytes that releasing in 14 days. That's right. Okay. The direct reason for the irregular menstrual cycle in a female and the occurrence of the disturbances in the secretion of this any disturbances it occurs in the females when the female not had a pregnancy. Maybe go to the doctors. Doctor advisable these females make analysis about follicle stimulating hormone and the LH hormone. Why? Because FSH and LH is stimulating the ovary to secretions, progesterone and estrogen hormone. So the main causes in that point, FSH and LH. So the best answer, FSH and LH, not estrogen and relaxin, not relaxin and FSH, not oxytocin and LH. That's right. In which of the following times for the beginning of the pregnancy is the sex of embryo can be determined? After 12 weeks, after 12 weeks or 13 weeks. So the best answer, after 3 months, 13 weeks, it is the best answer to can differentiations between male and female. Female, it's forming the ovary after 12 weeks, and the male, it's forming the testes and the hormone is secreting after 6 weeks. That's right. Okay? Which of the following contraceptive method it's preferred to the recommended when the pregnancy is very dangerous for the sick women? امرأة عندها مشكلة disturbances في السكشوال ريبرودكشنز عندها أو في الجينيتال سيستم عندها. advisable me coils, surgical operations, condom, pills, surgical sterilization. The best method in this case, surgical sterilization to prevent reaching the sperm into fallopian tube, not at all reaching. Why? Because it ligations it's occur inside the fallopian tube. That's right. Okay. Immunity in living organism. What is the mean of defense? What is the mean of defense to which the plant resort when it's failed in resisting the microbe that spread in some of its tissue? Hypersensitive response. When the plant is infected by the microbe and the pathogens, the infected cells must be destroyed completely through the hypersensitive response. So we can divide the method of induce methods that play an important role in the defense against the microbe and the microbe and the pathogen in the humans. Gums, cork-layers, cell walls, okay, cuticles, prevent enter the microbe and pathogens. Another type, prevent spreading. That means that microbes invasions, يعني دخل خلاص. So from that, cellular immunity structures surrounding the myceliums and the cell wall, and swelling is a part of cell walls. Tylosis prevents spreading the microbes. Formation of the parenchyma cells, it's swallowing and obstruct the bathing the waters and the pathogens, fungi, bacteria, virus. What else? Okay. Hypersensitive response, tylosis, okay, and hypersensitive response, tylosis, cellular immunity structure. So the best answers, hypersensitive response, not formation of the cork. Formation of the cork, prevent enters. Gum depositions, prevent enter. Formation in the uterus cover, prevent enter. That means that hypersensitive response, get rid of the damaged cells, so prevent spreading inside the healthy cell. That's right. The formation of tylosis is similar to the deposition of the gum in. Tylosis forming after the infections. Gums, it's forming when the plant cut. Yani that means that after the infections. So both of them is similar in shape and structures, wrong. Site of formations, wrong. C is the best answer. Why? Because the formation after the infection, both of tylosis and the gums is formed after the infections. Number D, preventing the entry of microbes. No, not at all, but the microbe already entered to the cells. Okay, that's right. Continuous with me, immunity in living organism. What are the chemical substances that may be used to inhibit the action of the hepatitis C virus in the liver tissue? Specific type of cells play an important role in the prevent spreading the virus, and Interferons is secreting a specific type of the protein. It's secreting from the infected cell with virus to prevent spreading the virus and avoid to infected the healthy cells. Interferons. Another types, TC, cytotoxic T cells. It secretes a specific type of the protein. Number one, perforin. Perforin make a perforated membrane of virus. And another types, lymphatic toxins, it is reaching to the nucleus. Inside the nucleus genes that found the infected cells. This chemical substance destroys the genes that found the infected cells, so destroyed completely the infected cells and to avoid infected the healthy cells. Okay? So the best answers, it is called interferons. The chemical substance is secreting from the chemical substance that's secreting from the cell infected with virus to prevent reproduction, the genetic material of virus. Not complementous. Complementous is associated with the antibody. Interleukin mediate communication between immune system and immune cells. Chemokines attract the macrophages in the site of the infections. That's right. To treat the person who was bitten by the poisonous snake, by which of the following substance is better for injecting him? Interferons related to the infected cell with virus, not related to antibody. It is the best answer. Why antibody? When the toxic substance enters to the blood, the antibody binding with the toxic substance and the complements enter of the group of reactions to reduce the toxicity of toxic substance found in the blood. So the best way it is the serum. It's called antibody that injected the human. It is the so the best way to inject the infected by the toxic substances that bitten by the snake with the antibody. Why? The complementers enter the group of reactions to reduce the toxicity from this point. That's right. Okay. فاصل قصير ونعود بكم إن شاء الله لاستكمال الحلقة النهائية لمراجعة الأحياء باللغة الإنجليزية. السلام عليكم ورحمة الله وبركاته. أعزائي طلاب الثانوية العامة شعبة علم علوم، نعاود مرة أخرى بعد هذا الفاصل القصير لاستكمال الحلقة النهائية لمراجعة الأحياء باللغة الإنجليزية. دمتم موفقين بإذن الله تعالى. Continuously about the immunity in living organism. What are the most abundant substances that are found in the tissue of an infected epidermis by the acne اللي هو حب الشباب اللي في الوش؟ What do you think? Chemokines, antibody, complementous, interleukins. Chemokines the best, right? Why? Because the chemokines attract the macrophages, the site infection. This acne that's found in the face, it's considered the site of the injury. So the microbes may be enter through this point. Okay? So the chemokines play an important role that attract the macrophages to engulfment of the microbe and the pathogens. Antibody? No antigen. Complementous is related to with the antibody. Interleukins is secreting from helper cells. Third line of defense, cellular immunity and humoral immunity. So the best line is the chemokines. Another questions about the immunity in living organism. What is the role of the interleukins? Activations B cells only? No. Activations helper and killer T cells? No. Okay. Activations B cells and helper T cells. The best right, it's called activations the B cells and helper cells. Why? When the B cells, okay, binding with the antigens through the receptor found in their membrane, inside the B cells, there is the MHC, major histocompatibility complex. When it's binding with the receptors with the antigens, the receptor found, the MHC antigen complex is formed in the surface of B cells. As soon as, at the same times, the macrophages engulf the antigens, and by the lysosome, it digests into small fragmented. The antigen is transferred into small fragment. Its size the macrophages as the same found in the B cells. MHC, major histocompatibility complex, is transferred on the surface, forming MHC antigen complex. The helper T cells, TH, it contains the receptor found in the membrane. It's called CD4. What that means CD4? Cluster differentiation force. It's binding with the MHC antigen complex and activating, secreting interleukin in the same times, binding with the B cells, MHC antigen complex in the B cells. The interleukins, it's found in the B cells, activation of the B cells to differentiations, plasma B cells, production antibody, memory cells, memory B cells, it's found in the blood approximately 20 to 30 years or maybe for lives. At the same times, in a cellular immunity, this binding it's occur MHC antigen complex, the helper cells. It is secreting interleukins. The interleukin activation the helper T cells against secreting cytokines, activations the humoral immunity and cellular immunity and productions memory helper T cells. So the best answers activation both of B cells and the helper T cells. That's right. Which of the following statement is correct about the natural killer cells? They are not specific. That's right, because it's found in the second line of defense and third line of defense. So it's not specific. The best answer, they are not specific for the certain type of antigen. They don't contain the perforance. They are the type of T cells. Okay, the type. Which of the following statement is correct? They don't contain perforance. They are type of T cells. No. T cells أنواعها TH helper T cells, TC cytotoxic T cells, TS suppressor T cells. Belong to the third line of the defense only? No, because it brings to the second line and third line. That's right. What happened when treating the bacteria S with the heat and the deoxyribonuclease enzyme, then transferring to bacteria strain Rs? Okay. To prove that the DNA genetic materials scientists divided into teams. التيم الأولاني قال إن البروتينز هو genetic materials. Why? Because it's specific type of 20 type of amino acid. But in DNA 4 type of nucleotide: adenine, guanine, purine, thymine, cytosine, pyrimidine. So the number of amino acid is more than the number of nucleotides. So the protein is a genetic material. That's wrong. Griff's experiment that's proof that the DNA genetic materials is strain Rs. It contains a gene forming the capsules in surface and out it. So it's called virulent strains. It causes pneumonia and lead to death. Why? Because it's coated themselves by smooth capsules. Okay. And another strain R, non-virulent, not rough, from outside that contains the gene that's secreting in the capsules. When injecting the mouse with the strain R, okay, the mouth is not die. And another strain S, the die because it's virulent. يبقى two type of strain causes pneumonia. Strain R, non-virulent. Strain S, virulent. When you injected the strain S that killed by heat and is injected with the strain R's, the strain R gains the genetic materials of the strain S and transfer from strain R non-virulent into virulent strain S, so some mice will die. When the infected injections, the strain S that killed and exposure to the deoxyribonuclease enzyme. Deoxyribonuclease enzyme decompose the genetic materials. No bacterial transmissions occur. Wow, the DNA not found. So it's called the crucial experiment. بيسموها التجربة الحاسمة. نحسم ان في دي ان اي وجيناتيك بكتيريا ولا لا، لان افري اند هيس كوليج قال لك الدي ان اي كونتامينيتد. وات ذا مين كونتامينيتد؟ يعني ملوث في كميه من البروتين، مي بي تكون الجيناتيك ماتيريالز. جت الكروشال اكسبيرمنت، جبنا الديوكسيرايبونوكليز انزيمز، انجكتد سترين اس كيلد باي هيت اند انجكتد with strain R. No transformation occur because the genetic material not found by decomposing the deoxyribonuclease enzyme. So the best answer D, the bacteria R will not be transferred into the bacteria strain S. That's right. Molecular purgy, according to the part of the DNA, the amount of DNA in the ovum of the female aphid insectus, it contains 2N. Producing of the female by parthenogenesis. As space, the amount is ovum aphid insects producing in the males is N. So the best answer is doubles. Why? Because the ovum 2N, it's to producing the natural parthenogenesis. Okay, natural parthenogenesis. 2n, it is producing in 2n without fertilizations by male gametes. Equal, that's wrong. Quarter, wrong. Half, wrong. That's right. What is the number of terns that are present in the piece of DNA double helix containing 1000 nucleotide? Okay. DNA double strand. Okay, double helix according to Watson and Crick. One run from prime 5 to prime 3 and opposite strand run from prime 3 to prime 5. Okay. Two strands always at the same distance from each other. Why? Because the type of hydrogen, because the type of nucleotide. In this strand, okay, the double ring may be guanine or may be adenine, and another strand thymine or cytosine, alternating with each other. So the distance between two strands always equals. The opposite directions, it's okay. One prime run from prime 5 to prime 3, and the other run from prime 3 to prime 5. Why? To give a proper correct about the hydrogen bond between the nitrogen species. يبقى الهيلكس ماسك في بعضه عامل زي الضفيرة, coiled around each other. Okay? Each ternus it contains 20 nucleotides. 10 in one strand, and the complementary strand it contains the 10. So the numbers of the double helix it contains 1000 nucleotides. It divided 20, so the total number 1000 divided 20, it's called 50 ternus. Okay? Number of nucleotides 50 ternus. Okay? Divide to 20. Okay? That's right. 1000 نيوكليوتايد المفروض هنا خطأ في الرقم. اوكي المهم نعرف نعمل divided اللي هو الترنس divided 20. اوكي divided 20. That's right. What is the number of nitrogen species that form two hydrogen bond with the base of the following strand in the DNA molecules? Cytosine, guanine, adenine, thymine, thymine, cytosine, adenine, guanine, adenine. Adenine connecting with the thymine with 2 hydrogen bond. Guanine with the cytosine with 3 hydrogen bond. What is the number of nitrogen bases 2 hydrogen bond؟ يبقى هنعد مين؟ الادينين والثيمين. الجوانين والسيتوسين مش معانا. طب نعد الادينين والثيمين؟ ادينين ثيمين يبقى دي 3. ادينين يبقى توتال نمبر 5. يبقى البيست رايت او البيست انسرز 5 because adenine connecting with the thymine with 2 hydrogen bond and the guanine with the cytosine with 3 hydrogen bond. That's right. Another questions. What is the number of nitrogenous bases pair in a part of DNA molecules that contains 150 terns? 150 terns by 20 يبقى التوتال نمبر كام؟ 3000. اوكي؟ each ternus it contains 2 nucleotides. 10 one strand and the complementary 10 another strand. Total number in ternus 20 nucleotides. يبقى التوتال نمبر كام؟ 150 by 20 equal 3000. هو عايز مني ايه؟ What is the number of nitrogenous base pairs؟ يبقى عايز pairs. هما 3000 يبقى البيز بير كام؟ يبقى 1500 يبقى البيست answers 1500 from DNA molecules. Okay. What does each chromosomes representing in the eukaryotics? There is a difference between prokaryotic and eukaryotic. Prokaryotic DNA in a form of plasmid, much smaller circular DNA, not constructed with protein, doesn't contain histone proteins. But in eukaryotics, زي أنا وأنت, DNA in a form of chromosome. Each chromosome contains annual molecules from DNA. Pay attention, please, in this point, very important. Before the DNA replications, chromosome found in 2 sister chromatids connecting from the center with the centromere. Each chromatide, it contained 1 molecules from DNA before the replications. After replications يبقى عندي في البروفيس والميتافيس الكروموسوم انتو كروماتايد. بروفيس ميتافيس انافيس انتيليوفيس السيستر كروماتايد. النورمال في الكروموسوم it consists only 1 chromatide it contain 1 molecules من DNA. Before the divisions the chromosome it consists of 2 chromatide connecting from the center with centromere. Each chromatide it contains 1 molecules from DNA. DNA coiled around the cluster of histone proteins. Okay, in the chromosome. shortened DNA 10 folds. It's called nucleosome. Nucleosome is specific only for eukaryotic. The replication it occurs and the transcription mRNA, rRNA, tRNA in a form of nucleosome. When the nucleosome is coiled, no replication it's occur. Why? Because the replication enzyme and the transcription enzyme doesn't reaching to the coiled. Okay? DNA coiled around the cluster of histone protein shortened 10 folds. The specific type of structure non-histone proteins shorten DNA 100,000 times, forming the chromatines. The chromatin condensing forming the chromosome. Okay? Chromosome may be 1 chromatide in normal rate, 2 chromatide for the cell division. Each chromatide contains 1 molecules from DNA. Okay. So the best answers: 1 molecules of DNA, a single DNA strand, that's wrong. 1 molecules of DNA, that's right. 2 molecules of DNA, wrong. 1 molecules of DNA and 1 molecules of RNA, that's wrong. Okay. Mutation appear on the offspring when they take place in the muscular tissue before the formation of the spermatogenesis or oogenesis, both of male and females. Primary germ cells قلنا على stem cells during the embryonic stage وهو الانسان في بطن امه سواء كان مال او فيميلز. يبقى الاساس الجامتس جايه منين؟ من البرايمري جيرم سيلز. يبقى لو حصل ديستربانسز في الستيم سيلز البرايمري جيرم سيلز يبقى الميوتيشنز ترانسفير تو انذر اوف سبرينج which called true mutations. Not transfers, false mutations. So the best answer: not muscular tissue. Skeletal cells of the testis. Skeletal testis found in the skeletal to keep the temperature below 2 degrees in normal rate اللي هي من 34 ل 35. Not related to. يبقى الإجابة الصح أو البيست أنسر primary germ cells. It is the mutations it's occur is transferred to the offspring. So the best answer C. That's right. What happened in a case of the absence of regulatory proteins from the chromosome? Regulatory proteins related to enzymes, hormones, and antibody. Regulatory protein. Structure non-histone protein related to creatinine found in nails, hoof, hairs, collagen that found tendons and ligament, myosin and actin found in the myofibrous main structures of the muscle, skeletal muscles. Okay, what happened in a case of absent regulatory proteins? Regulatory protein play an important role in formation the RNA polymerase enzyme, binding with the DNA, it's forming the promoters. Promoter on DNA, non-coding. After the promoter, the transcriptions, it's okay. So they play an important role in the formation of the promoters. What is the absence of regulatory proteins? DNA promoter can't be determined. Why? Because the DNA promoter is formed by helping enzymes and the regulatory proteins formation the enzymes. That's right. Okay. What is the first nucleotide triple that's follow the promoter on DNA during mRNA transcriptions? You know, the mRNA starting with the start codon AUG and the stop codon 3 UAA UGA UAG. يبقى انا عندي الستارت كودن AUG. يبقى الكومبلمنتري على التمبلت من برايم 3 لبرايم 5 ذاتس ترانسكرايبد فروم ات mRNA TAC. لو ترجمت التي سي هتبقى AUG اللي هو الستارت كودن اللي موجود على المين mRNA running from برايم 5 to برايم 3. Pay attention please. Focus in this point please اعزائي الطلبه. Transcriptions, translations, replications, 3 stage or 3 process it occurs or run from prime 5 to prime 3. Replications from prime 5 to prime 3. Transcription from prime 5 to prime 3. Translations from prime 5 to prime 3. Okay, that's right. In eukaryotics, RNA transcriptions occur in all the following cases except presence of regulatory non-histone protein in DNA. لسه قايلينها. That's right. Presence of DNA in the form of colon DNA nucleosome. لسه شرحها من شويه. No replications it's occur or transcriptions when nucleosome in form of coiled. Why? Because the replication enzymes doesn't reaching to the coiled nucleosome. Why? It's coiled around itself 100,000 times. يعني 100,000 مرة. على عكس النيوكليوسوم العادي shorten 10 folds 10 أضعاف بس. يبقى الإجابة الصح أو البيست رايت هتبقى presence of DNA in the form of coiled nucleosome. That's right. Which of the following representing a nucleotides triple for a stop codon on DNA strand. stop codons قلت لهم من شويه UAA UGA UAG لو ترجمت AA UUU ما ينفعش. B AGG not right تبقى UCC no. ACC تبقى UGG no. دي لا best right. Why? Because when the transformations in ATP on DNA templates the trans prime 3 to prime 5 it's transferred translations into UAA. That's right. Molecular biology, the following sequence present in the two segments of DNA X and Y studied in answers. X السيكونس من برايم 5 لبرايم 3 و3 من 5 والواي من 5 ل 3 ومن 3 ل 5. Which of the following is correct regarding this sequence? mRNA can't be transcribed from the segment? No. The segment X is containing more the phosphate groups at the same nucleotide at the same phosphate groups. Segment Y need to the high temperature to separating from the two strand. That's right. Segment Y it contains a large amount of guanine and cytosine. Focus in this point. Why? Because the guanine connecting with the cytosine with 3 hydrogen bond and the adenine with thymine 2 hydrogen bond. So the temperature is the 3 hydrogen bond it needing more temperature than the adenine to thymines. Okay, the gene whose effect is different in all the human being and can't be cloned except from its owners is located in the chromosome number 8. It's called gene. It's related to the fingerprint. And the chromosome number 9 it contains a blood group, a specific type of blood group. And the finally gene chromosome number 11 carry the gene responsible for the production of the insulin and the hemoglobin. Okay, thanks a lot for you. I wish I could see you again. Best wishes for you. إن شاء الله أتقابل معكم وأنتم في أحسن حال ومحققين أحلامكم. أعزائي الطلبة والطالبات دمتم موفقين في معية الله والسلام عليكم ورحمة الله وبركاته. كان معكم مستر حاتم منصور, biology teacher. Thank you a lot for you and goodbye.